Hello everyone, today's topic is on placenta. Placenta is a highly vascular structure. We can see here in the embryo. So we can see the placenta where it is connected uh, from the fetus by the umbilical cord. We can see the umbilical cord. So it is a highly vascular structure which is present along the unborn fetus and attached to the mother's uterine wall. This is the mother's uterine wall. The placenta consists of two components. One is the fetal component, the other is the maternal component. Fetal component develops from the chorion and the maternal component develops from the endometrium of the uterus. So the placenta provides the exchange of gases, nutrients and metabolic waste products between the mother and the embryo. So one thing to be noted that the placenta is the only organ in the body that develops from two different individuals. That is chorion from the fetus and the decidua basalis from the mother. That is the endometrium of the mother. So to understand the placenta development, it is important for us to understand implantation. So here we can see the fertilization occurs in the ampulla of the uterine tube. So after then the cells divide to form a 6-celled and 32-celled stage called as morula, and morula turns to form blastocyst which contains blastomeres and a cavity called as blastocele or blastocyst, and at the stage of blastocyst it implants into the wall of the uterus, that is into the endometrium of the uterus. So the process of implantation, it is a process by which the embryo is embedded and fixed into the endometrium of the uterus. So at the time of implantation, the embryo is in the form of blastocyst stage, and it usually occurs during 6th to 10th day after ovulation. So the time of implantation is 6th to 10th day after ovulation. The close contact of the fetal and the maternal tissues occurs by implantation of the embryo. So the blastocyst is surrounded by the zona pellucida, or which enters the uterine cavity on 6th day. The zona pellucida prevents it from sticking to the wall of the uterus, and as the blastocyst enlarges, the zona pellucida, which is the covering the blastocyst, it becomes stretched and ultimately it becomes disappears, and consequently the trophoblast cells, which is the outer cells, they are exposed. Now the trophoblast cells has the property of attaching itself to any tissue which it comes in contact with. So the trophoblast sticks to the uterine endometrium. The cells of the trophoblast divide mitotically and forms new cells that lose the cell membrane, and they form a jelly-like substance of mass of cells called as syncytial trophoblast. So here we can see the blastocyst having inner and outer cell mass. So the outer cell mass, once the zona pellucida is gone, the outer cell mass is called trophoblastic cells, which sticks to the wall of the endometrium. And here next stage we can see it is eroding the wall of the endometrium, and we can see the cytotrophoblast here, the cellular layer, and syncytial trophoblast, which is a jelly-like substance where the nuclear membranes are cellular membranes are lost, only nuclei are seen here. And these are the blood vessels present in the uterine endometrium, and here are the uterine glands. So the syncytial trophoblast, it invades the endometrium with the help of enzymes called as proteolytic enzymes secreted by its cells. So the syncytial trophoblast produces proteolytic enzymes which causes the erosion of the wall of endometrium. So the blastocyst goes deeper and deeper until it completely lies within the endometrium. So this kind of implantation in human is called as interstitial implantation. So the timing of the implantation, if you see, the implantation occurs during 6th to 10th day after ovulation in humans. And types of implantation, interstitial implantation, and in this type the blastocyst is embedded in the endometrium of uterine wall. And there are other types of implantations like eccentric implantation. Eccentric implantation is this type where the blastocyst is embedded in the uterine crepes. So within the crypt of the uterus it will go and embed. And example rat, which is called central implantation, and that is called as eccentric implantation. And lastly, the third type of implantation is the central implantation. In this type, the blastocyst is implanted in the uterine cavity. It is not in the wall of the uterus, in the uterine cavity. Example is the cow. So in humans, it is interstitial implantation where the blastocyst is completely embedded in the wall of the uterus. Sites of implantation, normal site of implantation is the upper part of the posterior wall of the uterine cavity. So the upper segment, anywhere in the upper segment in the posterior wall of the uterine cavity is a normal site of implantation. So abnormal implantation means if the implantation occurs anywhere else apart from the upper part of the uterus, it is called abnormal implantation. So abnormal sites of implantation within the uterus is in the lower segment of the uterus, near the internal os, and the abnormal sites outside the uterus can be like in the uterine tube, which is called as tubal implantation. Here we can see here, and in the infundibulum, it can occur in the infundibulum of the uterine tube, and in the interstitial part of the uterine tube, that is here. And also abdominal cavity, it can be outside the uterus and uterine tube fully. Within the abdominal cavity, that is called as abdominal implantation. And most frequently it can be implanted frequently in the peritoneal lining, like recto-uterine pouch or pouch of Douglas, and but may implant at any place covered by the peritoneum, that is mesentery. And it can occur in the ovary also, which is called ovarian implantation. So these are the various types of abnormal implantations. Normal site is posterior upper segment of the uterus. So fertilization and implantation may occur while ovum is still within the ovary in ovarian implantation. Next, about the ectopic pregnancies, occasionally the blastocyst implants outside the uterus, which is called as ectopic pregnancy. And like what we talked about, the uterine tube, ampullary and fimbriated end of uterine tube, then ovary, then peritoneal cavity in the abdominal cavity, and recto-uterine pouch or Douglas pouch. So they are all the examples of ectopic pregnancies. So usually such embryos, you find the death of the embryo if it is an ectopic pregnancy, and severe hemorrhage of the mother during the second month of pregnancy may occur. And rarely does an extra uterine embryo develops into full term. Actually it is very rare. Normally it won't develop at all. The normal place for a fetus to develop is the uterus. So suppose if the implantation occurs in the lower segment of the uterus, that is called as placenta previa. So normally we know the blastocyst implants along the posterior wall of the cavity of the body of the uterus, and developed placenta is attached to the upper segment. So wherever the implantation is there, that is the site of development of placenta. So as it normal placenta in the upper segment. So we see the normal implantation occurs in the upper segment of the uterus. So placenta develops in the upper segment of the uterus. So occasionally the blastocyst implants near the internal os of the uterus and then developed into a placenta which attached to the lower uterine segment, and the various degrees of placenta previa are seen here. So one is first degree is placenta previa, where the attachment of the placenta does not extend till the internal os. It is present somewhere in the lower segment of the uterus but does not extend till the internal os. Next is the second degree. The attachment of the placenta extends up till the internal os. You can see here, this is the second degree. It is extending up till the internal os of the cervix and but does not cover the internal os. Third degree is the most severe form, where the edge of the placenta covers the internal os, or when the os dilates the childbirth, the placenta is no longer occludes, but that is the third. Fourth degree is here, which completely covers the internal os. So the placenta completely covers and bridges the internal os. So when the cervix is fully dilated during childbirth, the placenta previa of the fourth degree is also sometimes termed as central placenta previa. And the placenta previa, especially the fourth degree, can cause a severe bleeding after the first trimester of pregnancy or during parturition, that is during childbirth. So that is the reason, because we know the placenta is a highly vascular organ, it is richly supplied by blood. So if this ruptures, it causes severe hemorrhage and the mother may die due to hypovolemic shock. So now let us understand the placenta parts of it. So we know the placenta develops from two entirely different sources, that is the fetal portion which develops from the chorion. So here we can see the chorion, which is a part of fetal portion. This is the chorion. This is the chorionic cavity or extra embryonic coelom. Extra embryonic coelom or chorionic cavity. This is the chorion, which forms the fetal part of the placenta. And the maternal portion of the decidua basalis will form the maternal part. So by the end of the third week, the anatomical arrangements are necessary for the physiological exchange of nutrients and gases between the mother and the embryo. So by the end of the third week, we see the beginning of uteroplacental association. By the end of the fourth week, the complex vascular network will form within the placenta, which facilitates the exchange of gases, nutrients, metabolic waste products between the mother and embryo. So it is highly specialized extra embryonic membrane that participate in the formation of placenta. And so we can see here, this is the extra embryonic mesoderm. So the extra embryonic mesoderm, we can see the cavity inside the extra embryonic mesoderm called as extra embryonic coelom. So the part of extra embryonic mesoderm is periphery forming the chorion, and the part of the extra embryonic mesoderm is along the yolk sac. So this is splanchnopluric extra embryonic mesoderm. This is somatopleuric extra embryonic mesoderm. And this is the uterine cavity, and this light color one is the endometrium, and the site of implantation, this part of the endometrium is called decidua basalis, and the rest of the endometrium which lines the rest of the uterine cavity is decidua parietalis, and part of the decidua which overlies the developing embryo like a capsule, which is called as decidua capsularis. So this is decidua capsularis. So decidua you can differentiate into three parts. Decidua basalis, which forms the placenta and it is the site of implantation. Decidua capsularis, which surrounds the developing embryo, Decidua parietalis, which lines the rest of the uterine cavity. So, numerous finger-like projections will form from the chorion, which are called as chorionic villi, and which we later discuss in the next slide as primary, secondary and tertiary chorionic villi. So here we can see the interstitial implantation. This is Decidua capsularis, which is covering it. Rest of it is Decidua basalis, and the site of implantation, rest of it is Decidua parietalis, and the site of implantation is the Decidua basalis. So after implantation, the blastocyst, that is the functional layer of the endometrium, is termed as decidua. That you have to note down. So the endometrium of the uterus is in the secretory phase of menstruation at the time of implantation. So as the embryo is implanted, the syncytial trophoblast starts secreting a hormone called as human chorionic gonadotropin hormone, which is otherwise called HCG hormone, human chorionic gonadotropin hormone, and which intensifies the changes that occurs in the secretory phase, and the stromal cells will enlarge, they get vacuolated and get filled with the till the brim with the glycogen and lipids, and this change in the stromal cells is called as decidual reaction. And change in the character of the endometrium after implantation of the embryo is now called as decidua. So we know the parts of decidua, which I already said. So decidua basalis is that part of the decidua which lies deep to the embryo where the implantation has occurred, where the developing embryo blastocyst occurs. And decidua basalis contributes to the development of placenta. Next is the decidua capsularis. It is that part of the decidua which forms the capsule around the embryo, that is the decidua capsularis, and separates it from the uterine cavity. Next is the decidua parietalis, that is the rest of the decidua excluding the decidua basalis and capsularis. It is termed as decidua parietalis. So let us understand the early stages of chorionic villi. So on the 9th day here, this is a schematic image for us to understand how the chorionic villi starts developing. So this is the decidua basalis, this is cytotrophoblast, and here is the extraembryonic mesoderm. Now there is a third layer forming, extra layer forming, which is the syncytiotrophoblast, which is derived from the cytotrophoblast, and this is the decidua basalis. Slowly lacunae starts appearing in the syncytial layer, which are called as maternal lacunae because they are filled with the maternal blood, and mother's blood vessels will be in the decidua, and syncytium starts eroding the mother's blood vessels, and slowly they start filling into this lacunae. So this is the first stage of uteroplacental circulation. So the lacunae gets enlarged, and here we can see still 3 layers. Let us understand how the villi forms now. So this is the decidua basalis, this is syncytiotrophoblast, cytotrophoblast, extraembryonic mesoderm. These are the lacunae. We can see mother's blood start pouring into the lacunae because of erosion of blood vessels, capillaries of the uterus. And in the next, this is how they appear. So here we can see the syncytium alone is present. The syncytium, if you take a cross section here, you will be seeing the syncytium surrounded by the pool of blood present in the lacunae. And later slowly the cytotrophoblast starts growing into the syncytium like a projection, which are called as primary villi. So this is called as primary villi. So here if you take a cross section, we see the cytotrophoblast surrounded by the syncytiotrophoblast, which is surrounded by the maternal blood in the lacunae. Now let us understand the development of secondary villus. Secondary villus means what happens, the extra embryonic mesoderm which is underlying this cytotrophoblast, the purple color one, it protrudes into the villi, turning the primary to secondary villi. So we can see this orange one is the syncytial trophoblast, yellow one is the cytotrophoblast, and purple one is the extra embryonic mesoderm which is protruding it. So if you take a cross section, it will be like this. The center part will be extra embryonic mesoderm surrounded by cytotrophoblast, syncytiotrophoblast, further surrounded by maternal blood in the lacunae. So this is secondary villi. Now let us understand the tertiary villi. Tertiary villi, we see proper blood vessels within the core of the villus. So we can see the artery and veins which are start developing within the extra embryonic mesoderm. So the capillary starts developing which are lined by the endothelium. So I will just tell, if you take a cross section, we would be seeing the endothelium lined capillaries surrounded by the extra embryonic mesoderm, further surrounded by cytotrophoblast, peripherally surrounded by the syncytiotrophoblast and the outer blood. So these layers will form placental barrier. So placental barrier are formed by the blood present within the capillaries is the fetal blood, and blood present in the lacuna is the maternal blood. So the layers between these two is called as placental barrier. So we will just see endothelium lining the blood vessel, that is the first layer, then extra embryonic mesoderm surrounded by cytotrophoblast, syncytiotrophoblast. These layers all together called as placental barrier. So here we can see the tertiary villi having placental barrier. So this is the maternal blood vessels, lacuna filled with the maternal blood. Lacunae are also having, this is the syncytiotrophoblast, cytotrophoblast. So now these villi, they branch and they anchor with the decidua, forming stem villi and anchoring villi. So stages of chorionic villi, if you see, initially the villi are formed all around the developing embryo. So all around the developing embryo, around the amniotic and yolk sac cavity, we see the developing of this chorionic villi in the decidua basalis. Later on, the syncytium will grow only at the site of implantation very much enormously, whereas the rest of the villi, they will just disappear and degenerate, making the surface smooth, covered by decidua capsularis. This part is called chorionic leaf because the chorion villi will leave from that place, so it is called chorionic leaf, whereas the other side is called chorionic frondosum. So a syncytial membrane, that is the chorion derivative, which is derived from the syncytium, the endothelial cells from the fetal capillaries. So human placenta is of hemochorial type. So the intervillus lakes is fully grown by the, if you see in a fully grown placenta, the intervillus lakes of this fully grown placenta contains approximately around 150 ml of maternal blood, which is renewed 3 to 4 times per minute. So the villus area varies from 4 to 14 ml and facilitating exchange of material between the nutrients and gases between the mother and the child. So as the chorionic space enlarges, the chorionic villi in relation to the decidua capsularis get compressed and degenerated, and as a result the part of the chorion becomes smooth and now it is called as chorionic leaf. So this part which becomes smooth, here we can see the chorionic villi, and this side there are no chorionic villi, which are called as chorionic leaf, which left the developing embryo. Chorionic villi in relation to the decidua basalis, that is here, grows extensively into the decidua basalis, and this part of the chorion is called as chorionic frondosome. This is called as chorionic frondosome. So this is the basic concept to be known. So here we can see chorionic villi all around the embryo, and here we can see they are concentrated only at the basal plate, which is called as chorionic frondosome. So here we can see initially the villi are present all around the developing embryo with the blood vessels. Later these villi will perish, only they are present at the site of implantation. So this forms the chorionic frondosome, this forms the chorionic leaf. So now we can understand the structure of a fully developed placenta. So lobulation of placenta. So after the formation of anchoring villi, so these are the anchoring villi. After the formation of anchoring villi, a number of septa from the decidua starts growing and grow inward from the uterine endometrium into the intervillous spaces, dividing the placenta into lobes. There are like 15 to 20 lobes, which are otherwise called as cotyledons. So there would be some 15 to 20 cotyledons. You can see the septa growing down, decidual septa growing down, dividing into the cotyledons. This makes the maternal surface of the placenta rough in texture because of the elevations formed by the cotyledons. So each cotyledon contains usually 2 to 3 anchoring villi. Here one is shown as a schematic diagram, but usually 2 to 3 anchoring villi. And as the pregnancy proceeds, the placenta also enlarges to meet the need of the fetus, and at the term the placenta covers nearly 30% of the internal surface of the uterus. So at term the placenta covers 30% of the internal surface of the uterus. So fully developed placenta, which is otherwise called full term placenta, is formed like a disc shaped. You can see it is like a disc shaped compact mass of vascular tissue, and it features like a flat circular disc, discoid, with the diameter of 15 to 20 centimeters. And usually it weighs around half kg weight, that is 500 grams of weight. So here we can understand the two surfaces of placenta. So placenta presents two surfaces, maternal and fetal surface. Maternal surface is rough because of 15 to 20 lobes, which are called as cotyledons, and fetal surface is smooth because of it's covered by a glistening amniotic membrane. And usually we see the attachment of umbilical cord almost approximately in the center of fetal surface. So this is the maternal surface, and this side is the fetal surface of placenta. So the cotyledons, they are so called because they appear to be like a cobblestones, or which are the rounded stones usually used to make the pavement in the gardens. So that is the reason they are called cotyledons. So we can see the presence of chorionic vessels running towards the chorionic plate deep to the amnion on the fetal surface. So here basic idea to understand a fully developed placenta.